Hi, and welcome to the second presentation in DNA synthesis. So the people who are most famous for working on DNA were these guys named Watson and Crick, and they were the people who discovered that DNA was a double helix or a twisted ladder. James Watson and Francis Crick were studying together at the university in Chicago. He ended up working at Cambridge with Francis Crick, who was a physicist. They were determined that they were going to be the first people to discover the 3D structure of DNA. Because scientists don't work alone, Watson and Crick also used the work done by Rosalind Franklin. Rosalind Franklin was using X-ray diffraction to study the structure of DNA. She discovered that DNA is helical and has a repeating pattern. Her work allowed Watson and Crick to come up with the model of DNA that we use today. They presented their findings in a paper in 1953, which was visually confirmed in 1969. So here's a picture of DNA as we visualize it today, a twisted ladder, deoxyribonucleic acid, or DNA. It's made of two complementary or two strands of DNA. It has a backbone that's made of deoxyribose, which is a sugar and a phosphate group. So the gray part are the deoxyribose sugar, and these orange and yellow things sticking out are the phosphate groups. The rungs or the rungs in the ladder are nitrogenous bases. They're complementary because they're not identical. Adenine pairs with thymine, and guanine pairs with cytosine. So A pairs with T, and G pairs with C. The bases are held together by hydrogen bonding. So here, the bases are being held together by hydrogen bonds. Base sugars and phosphates together are called a nucleotide. So if you take one unit of the base, which is brown here, the gray sugar and the orange phosphate, that's called a nucleotide. Deoxyribonucleic acid has a nitrogen base, so it's pointing to the thymine. It has a deoxyribose sugar, which is that brown ribbon. Attached to that brown ribbon are phosphate groups, and the bases are held together by hydrogen bonding. Here's another picture of a nucleotide, another type of picture. So we have our nitrogenous bases here, which is adenine. We have our deoxyribose sugar, which is this pentagon structure here, and the phosphate group are these three phosphates sticking out of the end. This is deoxy ATP, or deoxyadenosine triphosphate. So 1, 2, 3 phosphates, deoxyribose sugar, and an adenine base. This is a nitrogen base. So here's another drawing of a nucleotide. You have your nitrogen base, so adenine, thymine, guanine, or cytosine. You have your deoxyribose sugar, and you have your phosphate group sticking out of the end. Notice that the nitrogenous bases are attached to the sugar here and not the phosphate. DNA is a ladder where the nuclei or these nitrogenous bases are your rungs. A nucleotide is a repeating unit in your DNA molecule. DNA is twisted, so it's a twisted ladder or a helix. So here's a phosphate. Here is a deoxyribose sugar, and here is a nitrogen base. And notice that they're repeating patterns. So it's phosphate, sugar, phosphate, sugar, phosphate, sugar, with nitrogen bases in between. Notice that the other strand goes in the opposite direction. Sugar, phosphate, sugar, phosphate, sugar, phosphate, again with nitrogen bases. those nitrogen bases are held together by hydrogen bonds. That one repeating unit, base, nitrogenous base, sugar phosphate, is called a nucleotide. DNA bases come in two flavors. One are the big ones. They call these the purines. You can remember this by remembering that the bigger the molecule in DNA, the smaller the word. So purine is a short word for a double-ringed molecule. Pyrimidines are the smaller ones. They're the single-ringed molecule. So cytosine and thymine are pyrimidines, so they're small. Purines are guanine and adenine. Here you see that thymine is bonding to adenine. You can see here the hydrogen bond. This oxygen bonds with this hydrogen. This hydrogen bonds with this nitrogen. You can see that there are two two hydrogen bonds that are holding thymine and adenine together. And you can see that there are 1, 2, 3 hydrogen bonds that are holding guanine and cytosine together. The guanine and cytosine bonds are stronger than the thymine and adenine bonds. Again, you'll notice the hydrogen bonding. There are 2 between adenine and thymine, 1, 2, 3 between cytosine and guanine, 1, 2, 3 cytosine and guanine, 1, 2 adenine and thymine. We talked about how the two DNA strands go in opposite directions. We call this antiparallel. So you have sugar, phosphate, sugar, phosphate, sugar, phosphate on one side, and you have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar on the other side. So one strand runs antiparallel to another strand, so in the opposite direction. One strand is upside down. Here you have one hand in the opposite direction of another one, so that's antiparallel.